So let's say you run the job. Let's say it's an old job that you ran a long time ago. And then you see we have this new added feature. So this was a tree I ran earlier. I created this in Adobe Illustrator. This is a heat map view of presence and absence of protein families. And actually you can dig into that and find out different pathways that are the genes that are in these regions that are unique to one or the other are found in different pathways. And that's one of the things that makes these endosymbionts so extremely beautiful is they're evolving with their insect host. And if their insect host has a particular metabolic function that they both share, the endosymbiont will get rid of it. But generally, the way you become an endosymbiont is you give a special gift to your host cell and you give it a functionality that it doesn't have, and that makes it more likely to survive and reproduce and make more cells. Often those are in pathways. So that's what we're going to do next, is to look at not only the protein families, but the pathways. And I'm going to show you tricks that you can use or ways you can use the resource to find out what's unique about your data. Because this is what everybody wants, right? You've been working in the lab. You found these things. You think they're probably interesting because of some experiments you've done in the lab. You sequence them, you assemble them and annotate them. And really, we all need to publish to move forward, right? You can't put on your CV, I've sequenced 100 genomes. Give me a job. That's not going to happen. You need to get it into a publication. The better the publication, the better your CV looks, the better forward you move in your career. I mean, there are genome announcements where you can announce your genome, but those are not high quality publications. All of these tools are to help you find the things that distinguish your data so that the reviewer is going to say, oh, well, this is interesting, isn't it? Unfortunately, if it's, my new genome has 100 hypothetical genes that none of the other genomes have, you have to build a story, a compelling story. Not only are you scientists, I would say nonfiction writers, but maybe it's a little bit fiction writer, okay? You're creating a story to sell your data, not only to the reviewers, but to the community at large. And these tools hopefully will help you get there. You could even put this in a publication. You couldn't put it with all my little boxes saying it's this pathway that these guys have and this pathway that these guys have, although maybe you could. Might make a good publication. And these are the different insects that these guys were isolated from. So the last part of today, we've got one more thing to do. So let's click on Tools and Services. And this is something new. We have a new service that we have called Comparative Systems. So let's click on that. And you can add genomes individually. If I wanted to find my Wiggles, why do I keep doing this? Because it's so slow on this. Oh, it was pretty fast. So I could add my individual genome if I wanted to this. Why not? I'll just add it in. And then I can add those groups. So I'm going to use Racia test group and move that over. My own beloved, the 2 Wigglesworthia genomes, bring that over, and the Buchnera, and bring that over. Now, used to, on the Patrick website, we ran this, what they call is on the fly, so it would run here. And so I would repeat these things time again and again and again. But now we have it as a job so that you can go back into the services and see it. So I need my output folder, and then I'm going to name it Symbiont Comp Services. I'll just call it UVA Workshop. So it's that simple. And then I click Submit. Sometimes this is fast, sometimes it takes a while. But luckily enough, we have it in the workspace for your perusal. Let's go into the workspace. and go into BV-BRC workshop. And then I'll go into comparative systems. So click on that. And so I have my README and all that other stuff for it, but we want to see this job. So we click on that checkered flag and the job loads. And when you look at this, the downloads aren't that exciting. There's some JSON files and some TSV files. If you're a biologist, you never, ever, ever want to look at a JSON file. That's something that the computer scientists get all excited about, but we do not. We look at that and think, yuck. So the way to access everything is up here at the top. And you can see the pathways, the protein families, or the subsystems. Now, this tool is in the state of evolution. You guys are the first guys in a workshop to see this. Subsystems, we'll give it a try, but that part has been a bit wonky because we've never added it in before. But let's start with protein families with this icon. So click on that. And what that's going to load is the table that shows you there's a dynamic filter here and the results. So if I were to scroll down here, those are all the genomes that we used in that analysis. They're all there. And across those, there were 2,011 protein families. And you notice that it says PGF. These are the cross-genus family. And we have different genera here. So we need to do a PGF. When you're looking within a genus, you'd want to look at the local families, although the global families will work for you, but the local families are more fine-tuned and they'll be more exclusive. Like you could have 2 members in a cross-genus family when you're looking at something for your particular organism. But if you looked at it in the local families, the genus family, that might split it into 2 separate families. So you get a more fine-grained analysis. And up here, we have the genomes to the side. So what appears in this table is influenced by the filter here. In the first column, it tells you the ID for that protein family. It says that there were 6 proteins in that family in 6 of the genomes. This is what it was called. This is the minimum length from each of those 6 representatives. Here's the maximum length. And we also provide the mean and the standard deviation so that you get a sense of what's going on in that family and if it's a good one or not. A common thing when you're doing comparative genomics, things you'll see in papers are people report the pan genome, the core genome, and the accessory genome. The pan genome, this is the pan genome for this group. It's saying across all of these genomes, there are this many protein families. Over here, I can click on present and all, and I could do it individually, or I could click all of them. And then it says, oh, this is the core genome. For this group, there are only 3. Now, remember where we just saw those 3 before, where we saw 3 genes from the phylogenetic tree? So I could have done this first. If you have your genome or a group of genomes and you want to, maybe you just want to run the tree one time, maybe you want to see how many genes I can ask for. You could do this first to be able to see how many it's going to be able to find, right? Make sense? Okay, and then for accessory type stuff, like I could click on this. So we've got the Buchnera here. I could click on either present or either mixed, because right now I've said absent and all, and it says, okay, so I'm not finding anything because that's what you asked for. I could go through and click all of these for the Buchnera. And I could say, well, this is what the Buchnera have. This is the accessory for the Buchnera, what those Buchnera genomes have that none of the other ones have. Or I could look for an individual genome to try to see what it has that nobody else has. And that'll show me what that has. And I just was doing this ad nauseum the other day, trying to figure out and mapping onto a phylogenetic tree, because just like you guys, I'm trying to find something to report for this paper I'm submitting, and I'm trying to find if I can find proteins of interest to map onto this phylogenetic tree. So I have something big to say. I'd really like it to be like you can see with the insect endosymbionts, but for my own research, it's not quite that gorgeous. So those are the ways to get the pan, core, and the accessory genomes. I'm going to click on this again. You can filter here using names, which are problematic because like some of these names are just ridiculously long. And if you put it in there, there's a bunch of characters and I guarantee you it's going to be a mess. Or you could use protein family IDs. I use this one a lot. I work with Brucella a lot. And Brucella, there's a big distinguishing thing with these genomes as to whether they produce this lipopolysaccharide O antigen chain that sticks off on the outside of them. And the ones that are called the classic Brucella that have been known for a long time have that. But recently, we found that Brucella, they've been finding them in fish. They always thought that they were warm-blooded hosts. We found them in fish. We found them in frogs. We found them in a chameleon. And so one thing that differentiates them is the presence or absence of this lipopolysaccharide coat. So I have in my workspace for my private stuff, all of those protein family IDs, and I quickly put that into that filter box and filter it. And then I would know right away if they had it or not, or I could look at that. So that's the table. If there was a protein family I was interested in, I could click on that. I could do an MSA, and I hope it's not going to go away. It opens up a new page for that. So I could generate a multiple sequence alignment and scroll along it and look at the genes in that. And here's the alignment, and here's the gene tree. And this is just summarizing what you see in the alignment below. And I like it when it opens a new tab because I don't like it when it overwrites my page. So let's go into the heat map here. So click on this, and this is one of the parts I like the best. And I know you hear me say it all the time, and I know some of you are thinking, well, she says she likes everything the best. So sue me, I do. There are some things I don't like, but most of it I like a lot. So up along the top, it's showing you the key to this. Along this side are all the genomes, and along this side are all the protein families. I can grab the slider and bring it out, and you can see the protein families there. And when the cell is black, and if I mouse over it, you'll see that it's black. And in that pop-up window, it tells me the genome name, protein family ID and name, and it tells me that that particular genome has 0 members. So the black indicates nothing is there, right? Can't see it. If it's yellow, it means that one has 1 copy. If it's this tangerine-ish color, that means it has 2 or more copies in that protein family. And when glaringly orange, that means that there are 3 or more copies in that particular family. So if you wanted to search for things like, let's say I was interested in flagellar genes and I wanted to see where they were in here, I could start writing here and just look at that. I start writing flag. And across the top, those blue things, that's showing me the location of all those genes that have that particular text name within them. So that's another way you can search for things that you're interested in. If you do not like seeing your data this way, you can flip the axis. This is the way I prefer it. But now we're getting to the really good part. So this is all just higgledy-piggledy mix match of genomes and protein families. And it's matching what was in the original table. You can hit this cluster and it is going to rearrange the genomes and the protein families based on things that should cluster together. And suddenly things look a lot better, don't they? And another thing I can do is I can move things around. Like if I want to have this in phylogenetic order, I can grab these genomes and move them down. And so I can change this. And then when I have it just the way I like, if I want to create a great figure with this from this point, I can put the save here and I can say, save either this region or the entire thing as an SVG. Ta-da! How cool is that? And then look, you've got it ready for, Adobe Illustrator, and then you put the tree there and you just trim off that one and you have the tree there. And I mean, come on, admit it. Totally awesome. I know you all want to say it. I know you are just pretending to be tired and exhausted, but you're biting your tongue to keep from leaping to your feet and saying, hurrah! A question from the back. Young Clark Cuccinell has his hand up.
Could you just repeat the process of going back to the heat map, clustering, etc.?
Yes, and I can do that. It's going to keep the cluster though, so I'll just do a hard refresh. Bear with me because I know it's sort of like your favorite part of the movie when you just have to wind it back and see it again. This is what it's going to be like for you. So now it's just for pure pleasure. Okay, so I hit the table and then I'm going to do a hard refresh. So maybe it won't keep my clustering. Okay, so we're back there. I click on the heat map tab up at the top. And there it is. It's unclustered. It's a mess. It's like your closet. You open the door and all that stuff is there. And you think, if only I could be more organized, my life would be so much better. Well, you can do it clustering. And actually, there's an advanced cluster. So you could choose what you wanted to do. You can cluster by protein families, by genomes. We have different clustering algorithms. We have different clustering types, but generally I just stick with the basic the vanilla clustering, which is Pearson. So click on that and there you go. It's all clustered and you can download the SVG. But I know what you guys are thinking. You're thinking, this is just a picture. I want to see the data behind it. Is that what you were thinking?
I have a different question.
Okay, go ahead.
I was wondering, can you also add like 3 for proteins here?
You can add which proteins?
Like 3 for the proteins, like it's clustering.
Yeah. Well, it would be nice, right? If we provided it that way. I can arrange this in phylogenetic order right now. What I do, and I do a lot of work with Adobe Illustrator because that's where I generate most of my figures, is I put the tree there and then I line it up with the heat map, because that particular application is really good for me to do that. But right now we don't have it that way, but you should know because you just ran these things on a tree. So you can grab them and move them into phylogenetic order of that. So each of these can be moved and I can grab these things and move them around. When I get the picture that I want, I can save it, and it doesn't have to be all of this. It can be just a subsection of it. Like, let's say, come here, you. Let's say this is my favorite section, and I could even stretch it out more. Oof, if I wanted to. Sometimes you have to have a bit of finesse while you're messing with this thing. Now this would be my favorite part. So I could say, this is the one I want. It's perfect. That's what I want in my publication. I don't want the whole thing. So I click this and just say, save the snapshot. I've used this for years and I just actually found, I knew about this, but I hadn't really realized I could save the entire chart. So it was like Christmas. I didn't need anything else. All my desires were satisfied at that moment. A lot about bioinformatics, a lot of the time, the algorithms and stuff, they just overload you as biologists with data. And this is like, okay, I'm really interested in this section. So I start mousing over this stuff and okay, it's this gene. These are the genes that are unique. So what am I going to have to do? Am I going to have to get out my pencil and write down each of these things to search for them to find out more information about these things? Do you think that's what you're going to have to do? No, you are not, because we've solved that problem for you too. You use your mouse and you mouse over the section you are interested in. So I mouse over that and it's gathering the data that I selected from the heat map. I can download the proteins or I can hit show the proteins or I can add them to a group. Don't do that. Well, you can do it, but it's just a mix of stuff. So I can click on show those proteins and it's going to open up a new tab and it's going to show me what those proteins are. And, you know, I did it for a bunch of different genomes there. So it's going to be a bunch of stuff there, but it's all going to start. This is the genome ID. And all of it is him so far, whoever he is, up to here. This is all from this particular genome. So the one thing I really want to point out is a lot of these in the names here, you see a lot of EC with numbers after it. Who knows what that is? What is an EC number? Yes, it's the enzyme commission. And when you see enzyme commission, what's that an indication of to you? So it means it has a biochemical function. So it should be screaming pathways to you, pathways. Let's look at some of this stuff. When it's pathway stuff, let's see what the pathways amongst these organisms look like. We're leaving this behind because we're going to stop with protein families and we're going to move on to comparing pathways right now. Here we go. Go into workspaces, BV-BRC workshop, click on comparative systems, and then click on the endosymbiont comparison. I have to do this because if I go back or anything, I can't see my icons. So that's why I navigate that way. So up here we have pathways. So let's click on that. And this gives you something a little bit different. We use KEGG pathways. Does anybody know what KEGG is? Okay, that's the Kyoto enzyme, GG, whatever it stands for, I can't remember. So this is their pathways. And you can look at KEGG, Kyoto Encyclopedia of Genes and Genomes, and they have these kind of figures. And you know what? We have them too. It tells you the annotation. We used to also map the RefSeq annotation to this, but now we just have the Patrick annotation. This is the pathway ID. This is the pathway name. This is the pathway class. It tells me that there were 11 genomes had proteins in here. And this is the EC numbers, the gene count. So the EC numbers are each of the enzymes that they have that are in the pathway map. And there were 13 genes amongst these 11 genomes that had this. And there's the genome EC count. And there's one particular one that I wanted to show you that I like a lot. It was the histidine. So I clicked on pathway name to resort the table alphabetically. So I'm scrolling down to look at histidine metabolism. And unfortunately, remember, we're just bringing this into the new resource and improving it. We used to have a function where you could see how much it was conserved across the genomes in your selection. So if everything was 100%, that's not a pathway you want to look at because everything's good. You want to look at the bad stuff, right? Or the stuff that's not 100%, because that is where the gold is in your bioinformatic analysis. But we don't have that right now. So unfortunately, sometimes you have to click through all the results to find things that are interesting to you. Or if you knew the EC number and the name of the gene, you could go to it and see what pathways it was in. And I'll show you how to do that in just a second. I need to view this pathway. So when I selected the magical vertical green bar that shows me all the things I can possibly do, down at the very bottom, it has a map. So I click on that and it loads the pathway map and takes a minute for it to load. Still loading. It loaded the map, but it hasn't loaded the genes. Okay. So there are the genes and there is the map. And these things are sometimes quite large. So this tells me the EC numbers. You notice that there are cells that are colored and cells that are white. The ones that are white, these organisms don't have any genes that do that functionality. But these guys do. And if you look at the legend, you can see that the white ones are not annotated. These lime green ones mean that everyone has that gene. None of those are that lime green. They're all what we call this olive drab, and those are those grayish greenish things. Selected is when you have a group of genes and then you want to see it in the map, like let's say it's an RNA-seq analysis and you want to see what genes are in that pathway, then those would be blue. So you could see it from that. And red is select from the EC table. So let's say I want to see where this is the EC number, and I can scroll it out here. And that's the number of genomes that have that EC number. These are the number of genes across all of them that have it. And these are the genomes that don't have it. There are 12 that don't have it. And it occurs in the pathway twice. So if I click on that, it's going to highlight it so I can figure out where they are. Both tools, the comparative pathway view and the protein families by an analysis in Excel. I was given the task of having to do a comparative genomic analysis of this group of Brucella genomes. So it was before we had any of these tools in Patrick or BV-BRC. So it took me 3 months over the summer in Excel, assembling all this data. And across my wall in my office was littered these pathway maps with these kind of circles on them. And then I came up with all this data and the paper was published. And then I was part of Patrick at the time, but we didn't have this functionality. And our PI at the time said to the computer scientist guys, okay, make that into a tool. So they would come in and watch me work at my desk. It would be like they'd be sitting behind me and they'd be watching me do stuff in Excel. And they would say, why are you doing that? Because they can do it all on command line, right? So they could see how shackled I was by the current technology. So Malik actually, it took him like 2 weeks, I believe, to make the Excel sheet of it, just to do the Excel sheet that mirrored the heat map view. And then within like a month, we had the tool in the resource. So this is based on actual research.
Once we had the tool, within 30 seconds, you can get exactly the same figure
that it took me months to generate in Excel. Okay, so it's sort of like algebra.
All the tools that you see here, they are born out of suffering and misery.
Yes, and pain. So the question is, some of these organisms, they don't do much of these things. And the question is, does this mean that those things can't do histidine metabolism? Remember, these are the endosymbionts. Yes, that means they don't need to because their host cell is doing it for them. If they do it, that means their host cell has said, you know what, I don't have to watch this part of my genome anymore and regulate it to make sure that the replication is really good. If I have a mutation in this part of my genome, it's not going to kill me because my little buddy who's living in my cell is going to pick it up for me. Now, so the question is, okay, I can see that some of them have it and some of them don't. Well, which one is it? How do I find that out? And to do that, you click on the heat map view. And there it is. And you can see that the Buchnera all are providing this. It's important to Buchnera. It's important to their hosts that they provide this part of histidine metabolism. But Ricea and Wigglesworthia, their hosts don't need it anymore. So I'm sure their ancestor had it, but they've totally let it go. They don't need it. But these guys do need it because their aphids, Buchnera are in aphids, their aphid hosts are lacking that. And in fact, this was how the endosymbionts were found, was that Buchner was finding that when you put the insects at a particular heat, and it was high enough that it wasn't killing the insect, but it was hurting their endosymbionts, because this was back in the 1800s, suddenly these things started dying. And that's how he figured out that there must be something. I don't know if he even knew that they were bacteria at the time, that they were being killed off so they couldn't provide this to their host organism anymore. And that's why the host was dying. So you can see that you can use this as well to see that. Now, remember back when we talked about, because I don't have with the pathway maps, I don't have the thing that you're able to see which map is good or not. But when we were in that heat map view and I selected that group of things and I wanted to see which genes were in that some of them were missing. So it was on this feature table. And if it has an EC number here, I can click on that and then I can go to the feature view. So I click on that. And that takes me to the feature landing page, which will tell me which families, which KEGG pathways that's involved in. So this is another workaround that you can do in order to try to find the pathways that you want to look at. But come on, think about how many months this would take you to do, to assemble and annotate your genome and then build a tree and then try to figure out what's unique about it. So these tools are really there to help you. You're welcome to work on your own data. If you have data that you want to analyze on the third day and you want our help with analyzing it, if you have reads that you want to assemble, do that now. Assembly takes a long time. If you have RNA-seq data, go ahead and try to do that now so that the jobs will have time to run before that time. If you do it at noon on the third day and you submit your assembly and it's like, I'm here for you to help me, well, we'll just sit and stare at each other and maybe tell interesting stories about our lives, but we're not going to look at your data because we can't unless it's done. So go ahead and start your assemblies and your analyses, and we'll have it for the third date. That's all I got for now.
After going back, if you have some issues while doing the genome analysis, then you guys will respond, right?
Yeah, that's my job. We have had 12,000 of tickets. Sometimes we get as many as 30,000 jobs submitted every month. And when there's a ticket, meaning you had a problem, and you go into your jobs, my thing failed, you report that using the report issue icon. So click that. I use the report issue icon. That comes directly to me.
Okay.
Also, if you're up here in the workspace and you go into about and you hit contact us and you write in here something, whatever you want, that comes directly to me. I'm the first line of defense. I read those things. I try to solve it as much as I can because these guys are all doing improvements to the website, bringing in new services and things like that. And I don't want to disturb them unless I have to. So I take the first look at it. I try to figure out what it is. I try to answer your question, and then I alert them. Or sometimes if they don't have a lot to do, Bob has become very good lately at monitoring the ticketing stuff and going in and answering things before I even knew he had. So it's like a special little bonus that I didn't even know I was going to get. So yes, we will answer your questions, and we're contractually obligated to answer them within 48 hours, although some of us in this room are a little bit slower at that than others. And so when someone is slow, you just keep pinging it until you annoy that particular person so much that they must respond.